Hello everyone. Since from last few lectures we were discussing about mass spectrometry, this is once again from the same. As we know that mass spectroscopy is the most accurate method for determining the molecular mass of the compound and its elemental composition. In this technique, molecules are bombarded with a beam of energetic electrons and the molecules are ionized and broken up into many fragments, some of which are positive ions. In previous lectures, we have studied some ionization techniques. Now, in today's lecture, I am going to explain the atmospheric pressure chemical ionization and atmospheric photoionization. We know that several methods are there for converting the sample into gaseous ionic phase, that may be gas phase sources, desorption sources, evaporative sources, etc. The many ionization methods are available and each has its own advantages and disadvantages. Now, the function of ionic source is nothing but to convert the gaseous sample molecules into ions. And the energy that required for the conversion is different within the molecule. That means no single ionization method can be used for all kind of samples. Also, ionization method used depends on type of the sample under investigation and mass spectrometer which is available. Now this one is the classification. There are several methods which are available, and depending upon the nature of ions produced, that are classified on the basis of nature of substance and the method by which ions are produced, that are classified as gas phase ionization technique like electron impact ionization, chemical ionization, field ionization. Second, desorption techniques, that may be field desorption, matrix assisted laser desorption ionization, plasma desorption. Also the other methods includes electrospray ionization, atmospheric pressure chemical ionization, nanospray ionization. So few techniques we have studied in previous lectures. Now in today's lecture I am going to explain atmospheric pressure chemical ionization and atmospheric photoionization. So these few techniques we have studied in previous lectures: electron ionization, chemical ionization, electrospray ionization, thermospray ionization, matrix assisted laser desorption, FAB, fast atom bombardment, like that. In today's lecture, I am going to explain APCI and APPI. Ionization technique can be categorized into two parts, that may be hard ionization technique and soft ionization technique. Hard ionization technique, that means it includes the higher energy and number of fragment ions will be higher, but the number of molecular ions will be lower. And soft ionization technique, it includes low energy, low fragmentation, but higher molecular ion. Here, electron impact ionization, it is the type of hard ionization technique due to the high energy of electron impact. Now, let us see atmospheric pressure chemical ionization. Atmospheric pressure chemical ionization, it is soft ionization technique based on the mechanism of evaporation and carried out at atmospheric pressure. Actually, APCI is a combination of chemical ionization and electrospray ionization with some deviation. We will discuss how it is a combination of CI and ESI. APCI vaporizes solvent and sample molecules by spraying the sample solution into a heater which is heated to about 400 degrees Celsius using a gas such as N2 nebulizing gas. The solvent molecules are ionized by coronal discharge to generate stable reaction ions, and that ions produce that will go further to the analyzer. In APCI, the LC eluent is spread through a heated vaporizer at atmospheric pressure, typically at 250 to 400 degrees Celsius. The resulting gas phase solvent molecules are ionized by electron discharge from a corona needle. The solvent ions then transfer the charge to the analyte molecules through chemical reaction, which are also observed in chemical ionization. The analyte ions pass through a capillary sampling orifice into the mass analyzer. Generally, APCI, atmospheric pressure chemical ionization, is coupled with chromatographic instrument like HPLC, and here the corona discharge lamp is used for ionization purpose. Corona discharge needle or corona discharge lamp is used for ionization. Let us see the methodology present in atmospheric pressure chemical ionization. So these are few steps involved in atmospheric pressure chemical ionization. First one, HPLC eluent is passed through a pneumatic nebulizer where the droplets are both generated and desolvated. The spray so formed then passes through a heated region where the vapor is dried. Then the solvent molecules are ionized by a corona discharge to generate stable reaction ions, and here liquid flow rate is about 0.2 to 2 ml per minute. APCI, it is a variant of chemical ionization and is carried out using an ion source which is similar to ESI. APCI produces the ions using a reagent gas generated from the solvent vapor, where the solvent may be a mixture of methanol, acetonitrile and water at 0.5 ml per minute, which is supplied to APCI probe by a pump either by HPLC or by LC. Here, liquid spray is produced by passing coaxial nebulizer gas, which is N2, and the solvent spray is vaporized by a heating or by with the help of heating coils. Once that vapors are formed, that will be injected towards the mass analyzer. Now, the corona discharge lamp or corona discharge electrode is used for ionization, and the solvent vaporization, it is due to the heat nebulizing gas, heated nebulizing gas. APCI is analogous to CI as well as ESI. Why it is so? Here the corona discharge electrode is used for generation of ions that will produce the positive ions, and if we consider EI, on the atmosphere it also causes the electron removal from N2 O2, and that forms the molecular ions N2 plus O2 plus. In a complex series of reactions, the molecular ions react with H2O, CH3OH, methanol, and that forms H3O plus and CH3OH2 plus as reagent ions for Cr. So it is also similar with that of chemical ionization, and the H3O plus and CH3OH2 plus donate the protons to analyte, and it forms the ions like MH plus ions, which are called as quasi-molecular ions. So, corona discharge electrode will ionize solvent vapor molecule just like the production of primary ions in chemical ionization. Sometimes analyte vapors may also ionize by the electrode, and corona discharge electrode or beta particle emitted is used for ionization. Due to the collision and molecules into the ions, charge transfer take place between the solvent and analyte, so that there is the production of MH plus ions, which are called as quasi-molecular ions. Let us see the methodology in APCI. The first step is nothing but eluent. It comes from the column. Eluent, that is analyte plus solvent, comes out from the column and enters into a heated capillary tube. LC flow or HPLC flow, which contains analyte plus solvent. It comes out from the column and enters into heated capillary tube. The capillary is covered with nebulizing gas N2. Heated coil and nebulizing gas transfer the eluent into ionization chamber in the form of droplets. On droplets, the interaction of heated dissolution gas takes place. And leads to the formation of solvent analyte vapor. Corona discharge electrode or corona discharge needle will ionize the solvent vapor molecule just like the production of primary ions in chemical ionization. Sometimes the analyte vapors may also ionize by the electrode. Here the corona discharge needle or beta particle emitted that are used for ionization, and due to collision and ion molecules, charge transfer between solvent and analyte take place, and it will produce MH plus ions, which are called as quasi-molecular ions. So first step is molecules which are present in a gas phase. Then corona discharge needle ionizes N2 or O2 in source. Next one, that is N2 or O2 pass charge to the vaporized solvent. That passes the charge to vaporized solvent. Vaporized charge solvent passes charge to analyte. And the last one, the ions which enter mass analyzer aided by DP, that is by orifice. In APCI, the electric field is sufficiently strong to ionize solvent vapor by either removal or by donation of an electron. That means to form the positive ion mode or negative ion mode. Ion or molecule reactions then results in the formation of reactive species. Here, the acid-base reaction then takes place between the sample and reagent gas, which results in the protonation and forms the positive ions, which are called as the quasi-molecular ion, MH plus ions, or deprotonation, which leads to the formation of MH minus ion. The sample ions are then accelerated out of the atmospheric pressure source and the mass analyzer by application of a small voltage, typically 20 to 70 volt to the skimmer cone. The pressure differential between source and analyzer regions is maintained by the presence of an area of intermediate vacuum. During the ionization process itself, little energy is transferred to the sample molecule and fragmentation is minimal. So, APCI is soft ionization technique. So, atmospheric pressure chemical ionization, that includes ion evaporation and which is similar to chemical ionization. As here, corona discharge electrode will ionize the solvent vapor molecule just like production of primary ions in chemical ionization. Due to collision and the ion molecules charge transfer between solvent and analyte take place, and there is a formation of MH plus ions, positive ions called quasi-molecular ions. In some cases, there may be the formation of MH minus ion. So ionization occurs by two methods. When solvent ionizes, that leads to the formation of positive ions, and when analyte ionizes, formation of negative ions. For example, solvent ionizes, that means S plus A give the formation of MH plus positive ions plus S minus, and analyte ionizes where A plus S gives MH minus negative ions plus SH plus ions. In APCI, sample will be injected through a capillary, then it will be converted into sprayed droplets and finally analyte. So solvent vapor due to heating by N2 gas in the presence of atmospheric pressure, the vaporization and ionization occurs. Due to the collision, an ion molecule charge transfer between solvent and analyte take place, and that will produces the ions, positive ions and negative ions. By two techniques, solvent ionizes or analyte ionizes, where the solvent ionizes leads to formation of positive ions, analyte ionizes leads to formation of negative ions. So what are the advantages of APCI? It is the best applied to compounds with low to moderately high polarities. APCI may produce positive ions or negative ions. It is generally used to analyze polar thermostable substance, and also APCI having some disadvantages. Thermal degradation can occur, limited structural information as multiple charging do not take place, and it is not appropriate for higher molecular weight compounds. So APCI is analogous to CI, and for compounds with molecular weight about 1500 Dalton or less than 1500 Dalton, that will be used. APCI is generally used to analyze polar thermostable substance with molecular weight less than 1500 Dalton, and it also produces monocharge ions. Mostly when solvent ionizes, it leads to the formation of positive ions, and if analyte ionizes, that will leads to the formation of negative ions. Now let us see the second ionization technique, atmospheric pressure photoionization, APPI. So atmospheric pressure photoionization is performed at atmospheric pressure, hence called as APPI. Here also the LC eluent is vaporized using a heater at atmospheric pressure. The resulting gas is made to pass through a beam of photons generated by a discharge lamp, which is UV lamp, which ionizes the gas molecules. If we compare atmospheric pressure photoionization and atmospheric pressure chemical ionization, here only one difference. In APCI, there is a corona discharge electrode for ionization, and in APPI, there is ultraviolet lamp or photon generated lamp used for ionization. Now let us see the methodology. Here also the eluent comes from the chromatographic column. Eluent, that is analyte plus solvent comes out from HPLC or LC and enters into heated capillary tube. Enters into heated capillary tube. Then nebulizing gas and heated capillary tube forms the droplets of analyte and sample. Here also capillary is covered with nebulizing gas N2. Then heated coil and nebulizing gas transfer the eluent into ionization chamber in the form of droplets. On droplets, the interaction of heated dissolution gas takes place and it leads to the formation of solvent analyte vapor. APPI, atmospheric pressure photoionization. It is also a type of soft ionization technique based on the mechanism of evaporation. APPI is similar to APCI, but ionization in APPI is due to photons generated by UV lamp or UV light of krypton lamp. Whereas in APCI, there is presence of corona discharge needle. Here, the ultraviolet rays emitted from krypton lamp photoionize the vapor of analyte and sample. And the photoionization it takes place by three different mechanisms that may be direct APPI, indirect APPI and dopant APPI. So in atmospheric pressure photoionization, UV rays are emitted from krypton lamp and that photoionize the vapor of analyte and sample. APPI is coupled with chromatographic instrument like HPLC and here UV lamp are used to produce the photons. A discharge lamp generates photons in a narrow range of ionization energies. The range of energies is carefully chosen to ionize as many analyte molecules as possible while minimizing the ionization of solvent molecules. The resulting ions pass through a capillary sampling orifice into mass analyzer. Here also the dissolution gas N2 or heated gas N2 will be supplied which will convert the sprayed droplets into the form of vapors of analyte and the photons are emitted by the krypton lamp have the specific energy that is about 10 electron volts which is sufficient to ionize the target molecule that is to ionize the analyte and solvent. Photons of 10 electron volt will not ionize other atmospheric gas present due to lower energy. The photoionization takes place on the basis of three mechanisms. In APPI, photons will ionize the analyte by three mechanisms. These are as direct positive ion APPI, dopant APPI and solvent assisted positive ion APPI. That is direct APPI, indirect APPI, dopant assisted APPI or solvent assisted APPI. Let us see these two. Direct positive ion APPI. Here the analyte molecule M is ionized to a molecular radical ion M plus ion and the radical cation can then abstract an H atom from the abundant solvent to form MH plus ions which are quasi-molecular ions. So M reacts with H mu, these are photons, forms the radical cation and the radical cation can then abstract an H atom from the abundant solvent to form MH plus ions. Then second one that is indirect APPI where the formation of the molecular ions take place with the presence of solvent also. And here the abstraction of hydrogen atom from the solvent occurs to form these ions, MH plus ions, quasi-molecular ion. So direct positive ion APPI, one of the mechanism where the analyte molecule M is ionized to a molecular radical ion, and that will leads to the formation of MH plus ion by the abstraction of proton from the abundant solvent. Second one, dopant assisted or solvent assisted positive ion APPI. A photoionizable dopant or solvent D is delivered in large concentration to yield many D plus ions. Photoionizable solvent molecules achieve the same effect. Then these molecular cations, dopant cations, ionizes the analyte M by proton transfer. Again, these molecular cations of dopant ionizes the analyte M by electron transfer. That may be by proton transfer or by electron transfer. Here, dopant which is used for this process, that may be toluene, and toluene is used as a dopant agent to increase the percentage of molecular ion. So these are the few steps involved in atmospheric pressure photoionization. A vaporizer that converts the LC eluent to the gas phase, and the discharge lamp generates photons in a narrow range of ionization energies. What are the advantages of APPI over APCI? It is applicable to highly nonpolar compounds and low flow rates. These are other ionization techniques, or we can consider that is the summary of ionization techniques. Already we have discussed in detail electron impact ionization, typical analytes that will be very small or volatile, relatively small or volatile, sample introduction by GC or liquid or solid probe, and it is the hard ionization method provides the structural information. ESI, that is the soft ionization technique. MALDI, soft ionization technique, used for the analysis of peptides, proteins, non-volatile compounds. Fast atom bombardment, also used for the analysis of peptides, non-volatile compounds, and which is also the soft ionization technique. It is applicable to the mass range about 6000 Daltons, and the sample is mixed in viscous matrix like that. All these methods we have studied in detail. So in today's lecture we have studied two ionization techniques, APCI and APPI. In next lecture we will discuss some basic terms involved in mass spectrometry and also about fragmentation. Thank you.